Science. 15. Microbiology. The world of microbiology. Exploring the invisible kingdom of bacteria and archaea. Exploring the characteristics and diversity of these single-celled organisms, found in nearly every environment on Earth. Bacteria and archaea are two domains of microorganisms that exhibit incredible diversity in terms of their morphology, physiology, and ecological roles. Bacteria are abundant and can be found in diverse habitats, such as soil, water, and the human body. They play essential roles in nutrient cycling, decomposition, and symbiotic relationships. Archaea, although less well known, thrive in extreme environments like hot springs, deep-sea hydrothermal vents, and saline lakes. They are also found in diverse ecosystems, including soil and the human gut. Both bacteria and archaea have significant ecological, industrial, and medical importance. They contribute to human health, biotechnology, and the production of various commercial products. Studying the characteristics and diversity of bacteria and archaea is crucial for understanding the roles they play in ecosystems, their impact on human health, and their potential applications in various fields. Viruses. Unveiling the structure and function of these microscopic entities that can only reproduce inside the cells of living hosts. Viruses are infectious particles composed of genetic material, DNA or RNA, surrounded by a protein coat. They lack cellular structures and cannot carry out metabolic processes independently. Instead, viruses rely on host cells to replicate and multiply. They infect a wide range of organisms, including bacteria, plants, animals, and humans, causing a variety of diseases. Understanding the structure and function of viruses is essential for developing antiviral therapies, vaccines, and diagnostic methods. Viruses have also played a crucial role in molecular biology research, serving as tools to study gene expression, genetic engineering, and the mechanisms of cellular processes. Despite their pathogenic nature, viruses are integral components of ecosystems, influencing the dynamics of host populations and contributing to genetic diversity through horizontal gene transfer. Pathogenic microorganisms. Understanding microbes that cause disease, including their mechanisms of infection and ways to combat them. Pathogenic microorganisms, such as bacteria, viruses, fungi, and parasites, can cause a wide range of infectious diseases in humans, animals, and plants. These microorganisms have evolved various strategies to infect their hosts, evade the immune system, and cause damage. Understanding the mechanisms of infection, including adhesion, invasion, and colonization, is crucial for developing effective preventive measures and treatments. The field of medical microbiology focuses on the identification, characterization, and management of pathogenic microorganisms, playing a critical role in public health and infectious disease control. This knowledge enables the development of diagnostic tests, antimicrobial therapies, and vaccines to combat infectious diseases and protect human and animal populations. Antibiotics and antimicrobial resistance. Studying the use of antibiotics to kill or inhibit the growth of bacteria, and the growing challenge of resistance. Antibiotics are powerful drugs used to treat bacterial infections by targeting specific cellular processes or structures in bacteria. They have significantly improved human health and saved countless lives. However, the misuse and overuse of antibiotics have led to the emergence and spread of antimicrobial resistance, where bacteria develop the ability to survive exposure to antibiotics. Antimicrobial resistance poses a significant global health threat, as it reduces the effectiveness of antibiotics and makes infections more difficult to treat. Understanding the mechanisms of antibiotic action, the development of resistance, and strategies to combat resistance is crucial for preserving the efficacy of antibiotics and developing alternative treatment approaches. Microbial genetics. Delving into the DNA of microorganisms, including how they exchange genetic material and evolve rapidly. Microorganisms possess diverse genetic mechanisms that allow them to adapt and evolve rapidly. They exhibit various mechanisms of genetic exchange, including horizontal gene transfer, such as conjugation, transformation, and transduction, which enables the transfer of genetic material between different organisms, including different species or even domains. Microbial genetics explores how microorganisms acquire, maintain, and express their genetic information, leading to phenotypic variation and evolution. This field also investigates the mechanisms underlying antibiotic resistance, virulence factor acquisition, and the evolution of new microbial traits. By studying microbial genetics, scientists gain insights into the fundamental processes of genetic variation and evolution, providing a foundation for understanding the diversity and adaptability of microorganisms. Immunology, revealing the immune system's defenses against microbial invaders, including innate and adaptive responses. Immunology is the study of the immune system, which protects organisms from harmful microorganisms and other foreign substances. The immune system recognizes and eliminates pathogens through a complex network of cells, tissues, and molecules. It employs two main types of immune responses: innate immunity and adaptive immunity. Innate immunity provides rapid, non-specific defense mechanisms that act immediately or shortly after infection, while adaptive immunity generates specific responses tailored to particular pathogens. Understanding the immune system's mechanisms, such as antigen recognition, immune cell activation, and the production of antibodies, is crucial for developing vaccines, immunotherapies, and treatments for immune-related disorders. Immunology also plays a vital role in understanding the interactions between the immune system and microorganisms, including the strategies used by pathogens to evade immune responses. Microbial ecology, exploring the role of microorganisms in nutrient cycling, decomposition, and their symbiotic relationships with other organisms. Microorganisms are ubiquitous and play crucial roles in ecological processes, shaping the dynamics and functioning of ecosystems. They contribute to nutrient cycling by decomposing organic matter and converting nutrients into forms accessible to other organisms. Microbes are also involved in symbiotic relationships, forming mutualistic associations with plants, such as nitrogen-fixing bacteria, animals, such as gut microbiota, and other microorganisms. Understanding microbial ecology helps us unravel the intricate relationships between microorganisms and their environment, including the impact of microbial communities on ecosystem health, stability, and resilience. This knowledge has practical applications in agriculture, environmental management, bioremediation, and the development of sustainable practices. Probiotics and gut microbiome. Understanding the complex community of microorganisms living in our digestive tract and their impact on our health. The human gut harbors a diverse and complex microbial community known as the gut microbiome. This microbial ecosystem plays essential roles in digestion, nutrient absorption, metabolism, and the development and function of the immune system. Imbalances in the gut microbiome have been associated with various diseases, including gastrointestinal disorders, metabolic disorders, and autoimmune conditions. Probiotics are live microorganisms that, when administered in adequate amounts, confer health benefits to the host. They can help restore microbial balance in the gut and promote overall well-being. Studying the gut microbiome and probiotics is crucial for understanding the intricate interactions between microorganisms and the human body, as well as developing strategies to modulate the gut microbiome for therapeutic purposes. Fungi. Discovering the diverse world of fungi, including yeasts, molds, and mushrooms, and their vital role in ecosystems. Fungi represent a kingdom of eukaryotic microorganisms that encompass a wide variety of organisms, including yeasts, molds, and mushrooms. They are found in diverse habitats, from soil and decaying organic matter to human skin and internal organs. Fungi play critical ecological roles as decomposers, nutrient recyclers, and symbiotic partners of plants and animals. They are also important sources of food, medicine, such as antibiotics and immunosuppressants, and industrial products, such as enzymes and biofuels. Understanding the diversity, ecology, and physiology of fungi is crucial for ecological studies, biotechnology, and the development of antifungal treatments. Industrial microbiology. Learning about the use of microorganisms for the production of products like antibiotics, vitamins, enzymes, and fermented foods. Industrial microbiology applies microorganisms and their products in various industries, including pharmaceuticals, agriculture, food and beverage, and biotechnology. Microorganisms can be harnessed to produce valuable compounds through fermentation, bioconversion, or enzymatic processes. For example, antibiotics, such as penicillin, are produced by microbial fermentation, while enzymes derived from microorganisms are widely used in diverse industrial processes. Industrial microbiology also involves the development of genetically engineered microorganisms with enhanced capabilities for industrial applications. Understanding the physiology and metabolic pathways of microorganisms is crucial for optimizing their production potential and developing sustainable industrial processes. Microscopy techniques. Unveiling how microscopes, from light to electron, allow us to observe the microscopic world. Microscopy techniques enable the visualization and examination of microorganisms and cellular structures that are not visible to the naked eye. Light microscopy, including bright field, phase contrast, and fluorescence microscopy, provides insights into cellular morphology, dynamics, and interactions. Electron microscopy, including scanning electron microscopy and transmission electron microscopy, offers higher resolution and magnification, revealing ultrastructural details of cells and subcellular components. Advanced microscopy techniques, such as confocal microscopy and super-resolution microscopy, enable three-dimensional imaging and molecular visualization at nanometer scales. Microscopy plays a fundamental role in microbiology, facilitating the observation and characterization of microorganisms, their structures, and their interactions with their environment. Bacteriophage therapy, investigating the use of viruses that infect bacteria as a potential solution to antibiotic resistance. Bacteriophages, or phages, are viruses that specifically infect and replicate within bacterial cells. Bacteriophage therapy, also known as phage therapy, involves using bacteriophages to treat bacterial infections. Phages can kill bacteria directly by lysing them or indirectly by modulating the bacterial population and inhibiting the growth of pathogenic bacteria. Bacteriophage therapy has gained attention as a potential alternative to antibiotics, especially in the context of antibiotic-resistant bacteria. Studying bacteriophages and their interactions with bacterial hosts is crucial for understanding their therapeutic potential, developing phage-based therapies, and overcoming challenges such as bacterial resistance to phages. Biofilms. Exploring how some microorganisms form complex, surface-attached communities that are resistant to antibiotics. Biofilms are multicellular microbial communities that adhere to surfaces, encased in a self-produced extracellular matrix. They are prevalent in various environments, including natural habitats, industrial settings, and medical contexts. Biofilms exhibit unique properties and behaviors that differ from free-floating, planktonic cells. They are often more resistant to antibiotics, disinfectants, and the immune system. Biofilm formation is a complex process involving cell-to-surface attachment, biofilm maturation, and the development of spatially organized structures. Understanding biofilm formation and the mechanisms underlying their antibiotic resistance is critical for developing strategies to prevent or control biofilm-related infections, such as those associated with medical devices or chronic wounds. Vaccines. Understanding how these immune system stimulants protect us from microbial diseases. Vaccines are biological preparations that stimulate the immune system to produce an immune response against specific pathogens or their antigens. Vaccination has been one of the most successful interventions in preventing infectious diseases and has significantly reduced morbidity and mortality worldwide. Vaccines can consist of whole inactivated or attenuated microorganisms, parts of microorganisms, or specific antigens derived from microorganisms. When administered, vaccines elicit an immune response that generates memory cells, providing long-term protection against subsequent infections. Understanding the principles of immunization, vaccine development, and the immune responses they induce is crucial for disease prevention, public health, and global efforts to control infectious diseases. Microbial metabolism. Delving into the diverse ways microorganisms extract energy from their environment, including fermentation and photosynthesis. Microorganisms exhibit a remarkable diversity of metabolic strategies, enabling them to survive and thrive in diverse environments. They obtain energy through various processes, including fermentation, respiration, and photosynthesis. Fermentation involves the breakdown of organic molecules in the absence of oxygen, resulting in the production of energy-rich molecules like ATP and fermentation end products, e.g., lactic acid, ethanol. Respiration utilizes electron transport chains to extract energy from organic or inorganic compounds, such as glucose or hydrogen. Photosynthetic microorganisms, including bacteria and algae, capture energy from sunlight and convert it into chemical energy through photosynthesis. Understanding microbial metabolism and its regulation is essential for deciphering the metabolic capabilities of microorganisms, exploring their ecological roles, and developing biotechnological applications. Extremophiles. Learning about microorganisms that thrive in extreme environments, from hot springs to the deep sea, expanding our knowledge of life's potential. Extremophiles are microorganisms capable of living and thriving in extreme conditions that are hostile to most other forms of life. These conditions include high or low temperatures, high pressures, high salinity, acidic or alkaline pH, and environments with high radiation levels. Extremophiles are found in diverse habitats such as hot springs, deep-sea hydrothermal vents, salt flats, and polar ice. Studying extremophiles provides insights into the limits of life on Earth and the potential for life in extreme environments on other planets or moons. Extremophiles have also attracted attention for their biotechnological applications, as they produce enzymes and molecules with unique properties that can be useful in industrial processes. Microbial evolution and diversity, appreciating the immense genetic diversity of microbes and their pivotal role in evolution. Microbes exhibit incredible genetic diversity, reflecting their ability to adapt and evolve rapidly. They contribute to the overall biodiversity of the planet and play essential roles in ecological processes and the functioning of ecosystems. Microbial evolution encompasses processes such as genetic variation, gene transfer, mutation, and natural selection. Microorganisms exhibit various mechanisms for acquiring and exchanging genetic material, including horizontal gene transfer, which allows them to acquire new traits and adapt to changing environments. Understanding microbial evolution and diversity is crucial for deciphering the origins and relationships between different microbial species, studying the mechanisms of microbial adaptation, and exploring the evolutionary history of life on Earth. Emerging and re-emerging diseases. Exploring how changes in human behavior or environment can lead to the emergence or re-emergence of microbial diseases. Emerging and re-emerging diseases are infectious diseases that have recently appeared or have reappeared in populations, often with increased incidence or geographic spread. Several factors contribute to their emergence, including environmental changes, urbanization, deforestation, climate change, changes in agricultural practices, and globalization. Microbial agents responsible for emerging diseases can originate from animals, as in the case of zoonotic diseases, or arise from environmental reservoirs. Understanding the factors driving the emergence of infectious diseases and studying their transmission dynamics is crucial for predicting and controlling outbreaks, implementing effective prevention and control measures, and safeguarding public health. Soil microbiology. Understanding the role of microbes in soil health, nutrient cycling, and plant growth. Soil is a complex ecosystem that harbors an immense diversity of microorganisms. Soil microorganisms play critical roles in nutrient cycling, decomposition of organic matter, and the formation and maintenance of soil structure. They contribute to plant growth and health through various mechanisms, including nitrogen fixation, nutrient mobilization, and protection against pathogens. Soil microbiology encompasses the study of microbial communities in soil, their interactions with plants and other organisms, and their functional roles in soil ecosystems. Understanding soil microbiology is crucial for sustainable agriculture, land management, and the development of strategies to improve soil fertility, enhance crop productivity, and mitigate environmental impacts. Infectious disease outbreaks and pandemics. Investigating how microbial diseases spread in populations and measures to control them. Infectious disease outbreaks and pandemics are characterized by the rapid spread of microbial diseases within human populations, often leading to significant morbidity, mortality, and societal disruption. Understanding the dynamics of infectious disease transmission involves studying factors such as pathogen characteristics, host susceptibility, modes of transmission, and the impact of social, economic, and environmental factors. Epidemiological research plays a crucial role in investigating and controlling infectious disease outbreaks, informing public health interventions, and developing strategies for disease surveillance, prevention, and preparedness. Antiseptics and disinfectants. Learning about substances that kill or inhibit the growth of microbes, crucial for preventing the spread of infections. Antiseptics and disinfectants are chemical agents used to kill or inhibit the growth of microorganisms on surfaces, skin, or mucous membranes. They play a vital role in preventing the spread of infections in healthcare settings, public spaces, and everyday life. Antiseptics are typically used on living tissues, such as skin, to reduce microbial populations and prevent infection. Disinfectants, on the other hand, are used on inanimate objects and surfaces to kill or inactivate microorganisms. Understanding the properties, modes of action, and appropriate use of antiseptics and disinfectants is crucial for effective infection control, ensuring patient safety, and maintaining a hygienic environment. Thanks for watching. Please like, share and subscribe, and comment below if you agree, or would like to add your thoughts.